Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Adam Nelson, Chromatography Product Manager from Repligen.
Welcome and thank you for joining. My name is Adam Nelson, Chromatography Product Manager, working on the Opus Pre-Packed Chromatography Column product line at Repligen. I'm here to talk to you about the impact of statistical process control analysis when looking at column packing data and how that can relate to a reliable and consistent and predictable performance during your purification using Opus columns. So before I jump into that, I would like to discuss how Repligen fits within the industry and how we take a different approach within the bioprocessing industry. So Repligen provides products all through the product stream here for customer processes, upstream, downstream, analytics, flow paths, systems, supplying products into new modalities, new projects, and with a focus on being an innovative leader, working with our customers to have a flexible design of products and to deliver a custom and customized solution. We are focusing on being first to market and supplying products that will basically improve, process efficiencies for our customers. One of those leading product lines from Repligen is the Opus Pre-Packed Chromatography columns. As you can see here on the slide, we are offering a variety of diameters, starting with our smallest scale here, the process development columns, namely the Robo column, Mini Chrome column, and Valley Chrome column. But we also have options that scale harmoniously and with a design into pilot scale, all the way up into manufacturing commercial scales. So starting at a small scale with Opus columns, you have the confidence of a harmonized design as you scale up through pilot scale, commercial phases, and potentially all the way up into your 80 centimeter columns. What separates Opus from other chromatography columns is the ability for our customers to specify the diameter, the bed height, and the resin type being packed. Repligen will study and pack any of the resins out there from the leading vendors for packing into your columns. So our customers have the choice to design the column and specify the criteria for acceptance. Putting all those three factors together, diameter, bed height, and resin, we pack the column and deliver a product to you with a quality package, so it's ready to go for easy onboarding at your site. So as you look at the variety of diameters here, it took quite a long time to get to this point right now. So it all started in 2012 when we launched the Opus 20 column and Opus 30 column. That entered the market, and we had great feedback, and we were listening to our customers, listening to the industry, and saw that we needed to expand the offering. So as you go through the years, where we were adding sizes smaller than 20 centimeter, larger than 20 centimeter, to really build out the full portfolio for you. So namely, 2016, adding Opus PD columns into the portfolio, increasing the sizes 45, 60, 80 centimeter columns through 2017 into 2019 for those manufacturing commercial scale processes. Also even adding the Opus 2.5 in 2021, and this year adding Opus 36 into the portfolio. When you look at this timeline, this allowed us to gather thousands of columns worth of data with the variability of bed height, diameter, and and resin type, Repligen is in a unique position to take that information and use it for continuous improvement within our process engineering team and our manufacturing team. So taking all of these factors together in terms of deliverables for an Opus column, the harmonized design throughout the scaling of the column sizes, the documentation that comes with your column, qualifying that for easy onboarding, and the thousands of data points, as the backbone of our process excellence in packing your columns. You're not only getting a product, you're getting a reliable performance that has consistency and predictability for your operation when using it in a purification. Digging a little bit more into those data points that we have for our packing methods and testing methods, here's a representative trend here for asymmetry and efficiency. So we took thousands of those columns in over 250 resin types and all different applications, and we're able to leverage that data to understand our process, make process improvements, and therefore improve the product for our customers' use in their applications. We've been tracking this data all along for over 10 years, and now we have an opportunity to take that data and share it with our customers for improved understanding of process controls and how that can relate to column performance. So our strategy here was to look at a variety of factors when looking at the data, namely the resin type, categorizing our methods based on resin type, and seeing how column size, some decisions on bed height or diameter, or changing both, how those can change the process data that we collect. We are focusing statistical process control analysis to our efficiency and asymmetry, which are the core parameters for evaluating a well-packed column. Then we take that data and see how things improve as we implement process changes accordingly. So diving into the cycle of continuous improvement that we use, it all starts with gathering the data. So for the first 4 to 5 years of packing columns, we gathered data to the point where now we have a substantially significant size of data set to analyze. So we put that into our process control charting methods and software that leverages data variance and standard deviations to apply a control score. So it looks at the variability of that data and normalizes everything to a score. When we look at all of our scores, we find some resin types that are a little bit lower than average. We'll focus on those in the process engineering team and the R&D team to evaluate the packing method, see if there's something specific we might change because of a certain diameter or a certain bed height within that family of resins. So we'll go through that packing study, send it back into our production team as a tech transfer, and have them implement that slightly changed process and see how our scores improve. As we iterate through this system of plan, do, act, check, repeat, we get to a point where now we have hundreds of resins that are as established as well controlled in our packing process and as a standard offering to our customers. So this is the power of continuous improvement, which we implement within our manufacturing site for the past 10 years. So going through all of that data, it was improving our internal processes in terms of packing consistency. But now we have such a large data set and an opportunity to share this with our customers because of the variety of column sizes we're packing, the variety of resin types we're packing. We have thousands of different configurations. But here to illuminate on that is an ion exchange resin here that we ran through our statistical process control analysis. This is a family of resins with a consistent backbone, but all ion exchangers. What's to note here is that it's a single resin family, but we have variety of diameter and bed height represented here. So we have 1,000 individually packed columns between the range of 5 to 80 centimeter in diameter. And looking at the top chart here for reduced HETP, this is our measurement of efficiency. We see some clear bands here for the upper control limit and lower control limit. Obviously, there's some variability in the data above and below, but looking at the standard deviation, over 95% of the data is falling within this range and would be deemed as an acceptable value here. So we have a greater than 95% confidence that the reduced HETP will fall between 2.5 and 4.5 to show a well-packed column. Typically, we're accepting of something less than 5 here as a standard to the industry. Specs being maybe less than 7 on the reduced HETP. Looking here on asymmetry, we see a similar trend here that some are above the line, some are below line, but greater than 97% of the results are going to be within that 2 sigma of standard deviation here. So you have a confidence that you're going to receive a consistent column, and that will then translate into a consistent performance on your end. This is a very impressive data set here because it's capturing data from over 8 years of packing and 1,000 data points here. So very significant data set to show that process controls lead to consistency and performance here. Taking this further, we wanted to show how our method of continuous improvement has actually changed the data slightly. So originally, when we started this program in 2014 of gathering the data, analyzing the results, we had a baseline average reduced HETP here of 3.9. And then in implementing in 2018, we switched to a different packing method, dynamic axial compression, leveraging the top flow adapter to compress that resin. Provides a much more consistent and repeatable process for us. And we saw an improvement here. So a reduction of reduced HETP is an improvement in efficiency here. So we saw that improvement. And in reference to the top right chart here, we also saw a tighter distribution of the data set. So we are having a better grouping around that average value as well. And so that's really showing the consistency here in a quantitative way. Moving further into 2019, 2020, we implemented skid automation. So, using our built methods within for packing and testing within our skid to give that reproducibility, remove the human factor from the setup and the execution of the test. We saw an improvement on efficiency. Finally, since 2020, we've been seeing a lot of orders and from a bundled strategy where a multi-column order would come in. Maybe it's 10, 15 columns from the same customer, same order, same configuration for diameter, bed height, and resin type. And that allows us even more process efficiency here and an improvement to the reduced HETP value. So over the years, we're implementing the continuous improvements and adding those improvements into our process. And you see that in the quantitative data here, improved our reduced HETP value and a much tighter distribution of the data set over the years. Digging deeper into the data set, you may wonder, what happens when we look at the different segments of diameter, right? So let's break down the same data set into small diameter, medium diameter, and large diameter. When we break that down, we still see that variability of values that are higher or lower than the average here. But overall, we're in a really strong band of performance from 3.3 on the reduced HETP up to 3.8 in the range here for the medium. So even if you are setting your expectations at the small scale, let's say 5 to 10 centimeter diameter column, you have the confidence that it's going to scale for you. The same expectation in the Opus columns is going to be there when you go up into that 45, 60, or 80 centimeter diameter point of your process. So there is that reproducible performance for you as you scale. Taking this even a little bit further, if we were to really shrink down the data again to a singular resin type, in this case, we chose an affinity resin type with a single diameter being chosen and bed height. So this is a unique configuration. You see a much tighter distribution, a nice grouping of the data when you look at the first line here for the smalls, asymmetry and reduced HETP. A nice grouping of data, and this is achieved through consistency of request on the bed height, the diameter, the resin type, and the specifications used to test the column. So over the years, we have established standard packing and testing criteria for our resins. And in this case, this reproducible order is leveraging that. All of these columns are using the Repligen standard specifications for testing the column. So with that process consistency, column sizing consistency, and that reproducible order, we see this nice grouping of data. Scaling that same resin type into a larger diameter column, in this case, we have a 45 by 25 centimeter column being represented. We still see that nice grouping even with the scaling, much less data points that you compare it to the 5 centimeter here, but still a great grouping so that you have that confidence of scale up regardless of the size of column. And then taking that data, we saw that the need for reproducibility from order to order from one year to another, or within a particular order of multi-columns was really really important for emerging markets in gene therapy, right? Where multiple columns are being used to represent multiple dosage, multiple purifications for the product. There is that requirement of reproducibility as each column needs to be the same to represent the population of the study there. So the same thing applies here when we show our SPC analysis. A nice tight grouping here, looking at a small scale column with an AAV affinity resin. We have greater than 98% falling within the 3 sigma and meeting our specification here for efficiency, and then 99% within the 3 sigma acceptance criteria on asymmetry. So whether you're ordering column number 50 from this data or column number 350 from this data, you have that consistency of the performance coming out of the manufacturing side of it, which then leads to your confidence in using it in your programs. So to summarize all of this, going the route of Opus columns simply takes away the variability that you may have in packing columns. We can leverage the thousands of columns worth of data to pack the column for you and eliminate the packing risks from your side so you can focus on the value-added steps of actually purifying your product. When implementing Opus columns and leveraging the data for this scalability and consistency of performance, you have optimized project timelines because you can go right into your production with the confidence of that background data supporting the decision. And then finally, putting all of this together, it's all about increasing the process consistency here. So once you establish a performance with your first column or your first the set of 5 columns, you know that the next set is going to be there from a performance standpoint, leveraging our Opus columns with our standard specifications for packing and testing. That will translate into a consistency of performance during your purification. Finally, that ends the presentation here. I'd like to thank you, and I'd be open to any questions you may have. Thanks again.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, What parameters are open for configuration or customization on Opus columns, and how would that impact your dataset?
Yeah, so as shown in the presentation, the key features to configure are the choice of resin, bed height, and diameter. But our customers also have the possibility of choosing the solutions used for testing, the injection volume, the testing flow rate, as well as the storage solution of your column. So in particular, those testing parameters of the combination of injection solution type, volume, and testing flow rate is going to heavily change the target for asymmetry and efficiency. And so that's why having a consistent process of testing your column is going to give you the best opportunity to evaluate the consistency of performance of that column.
Okay, and our next question is, do you offer a document that lists out the Repligen standard specifications that are referenced? Solutions, flow rate, asymmetry, etc.?
Yeah, so we have a database for our internal use for reference in the quoting discussion when talking about placing an order for Opus columns. We haven't created that. It's a very large list, so we haven't created that. But as requested, we can send over what our standard would be in conversation with your local sales member.
Okay, and is it possible to sterilize those columns?
So we wouldn't claim an ability to sterilize. We sanitize columns, but there is an option for possibly for gamma radiation of columns to show low bioburden, but we wouldn't have a sterility claim on chromatography columns at this time.
All right, and our next question is, do you have recommendations for post-use storage of Opus columns, specifically the way to store the column hardware, not which chemical to store in?
Sure. Yeah. So as developed, the product line shows great stability from a construction standpoint at low temperatures, right? That 2 to 8 degrees, as well as higher temperatures, 40 degrees. Our suggestion for storing the columns obviously is in a safe space, right? We don't want to be in a high traffic area, but otherwise, from a temperature standpoint, it's dependent on the resin type. So the resin type guidance will guide you towards the temperature for storage of your column as well.
Great. Our next question is, how would column performance specifications change with scaling up in diameter?
Right. So in a review of our data, we saw a correlation of diameter and bed height, that really didn't change the results that we're seeing here for efficiency and asymmetry. But one thing to keep in mind is that you will see a slight change in pressure and flow rate. So in that way, a scale up needs to consider a change in pressure and flow rate, but relatively the target for efficiency and asymmetry is going to be roughly the same as you're scaling up with Opus columns.
All right. Our next question is, can you explain the gamma irradiation of columns post-packing? Does this include the resin?
Yes, it does. It's a packed column that we would send out for gamma radiation. So that is something that we would launch on a larger scale later this year.
Awesome. And how does bed height play a part in the data consistency?
Sure. Right. So as shown in the presentation, there is an effective bed height here, right? So and it depends on the resin type. Some of the smaller particle resins, when you're packing to higher bed heights, you're going to see that much more variability, just because of the flow properties going through the column. But within the range of about 10 to 20 centimeters, which is the majority of the packs that we're performing, there really isn't going to be a variance there. When looking at the edges, maybe closer to 30 centimeter bed height or 5 centimeter bed height, when the flow properties can change quite a bit, going from the top flow distributor to the bottom flow distributor. There can be a slight shift there, and we could certainly discuss the targets if those opportunities come through for your projects.
And how is the flow distribution reproducible across the different column diameter?
Yeah, so that's a great question. We've taken quite a bit of time to design our flow distributors to promote the flow of liquid from the radial center of the flow adapter to the radial edges before pushing through our mesh and into the packed bed. So there is a special design and features within the flow adapters to promote that, so that we do have a nice even laminar flow through the column, and it's being collected evenly going out of the column as well.
And assuming the availability of resin, what is your turnaround time for packaging columns on average?
Yeah, so on average, we've been really solid at about 6 to 7 weeks from the resin availability within our production suite, with certainly enough capacity to help grow and keep that maintain that lead time of 6 to 7 weeks from the availability of resin.
All right, great. Well, it looks like we're running a bit short on time, so if we don't get to your questions, we'll be passing them along to Adam, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. So the last question for the webcast is, what controls should end users put in place for their own testing to reach the level of consistency presented?
Sure, yeah, so certainly there are some external features to the column that should be controlled. The method of injecting, the tubing setups in terms of length of tubing, diameter of tubing, this should all be very consistent. And our suggestion is to match the parameters from our C of A. Each one of our columns is delivered with an individualized C of A outlining the testing parameters. So following that is a good rule of thumb. But in general, making sure that the pump is consistent, the setup is consistent, your flow rates are consistent, the placement of injection, the placement of conductivity meter are consistent. These are going to lead you towards a much more informed decision that a column is going to be good for your use or still needs some investigation. So that consistency of the setup and the method is going to drive that consistency of your performance.
All right, well, fantastic. Thank you so much, Adam.
You're welcome.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to have you joining us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Bye.